Bioluminescence, the ability of living organisms to produce light, is a fascinating natural phenomenon. At the heart of this process are two key molecules: luciferin, the light-emitting molecule, and luciferase, the enzyme that drives the reaction. These two components work in harmony to create the glowing displays we associate with bioluminescent organisms. Let's dive into the details of how luciferin and luciferase interact and their importance in nature. Luciferin is a light-producing compound found in bioluminescent organisms. It acts as the fuel for the bioluminescent reaction. The term luciferin comes from the Latin word lucifer, meaning light bringer. Characteristics of luciferin: it is a relatively small organic molecule. Different organisms have different types of luciferin. For example, fireflies, jellyfish, and certain types of plankton use structurally unique forms of luciferin. It is highly reactive with oxygen, a key feature that enables the production of light. In essence, luciferin is the molecule that gives bioluminescence its energy and color, depending on the specific chemical structure and environment. Luciferase is an enzyme that catalyzes the reaction between luciferin and oxygen. It acts as the match that ignites the light-producing reaction patterns of a sponge. As part of the action associated with luciferase, it is a protein specifically adapted to interact with luciferin. Different species have distinct types of luciferase which work with their specific luciferin molecules. Luciferase is highly efficient, allowing organisms to produce light without generating much heat, a phenomenon called cold light. Luciferase ensures that the reaction proceeds quickly and efficiently, enabling organisms to produce light on demand. The process of bioluminescence involves a chemical reaction between luciferin and oxygen catalyzed by luciferase. Here's how it works. Luciferin activation. Luciferase in the presence of oxygen molecule binds with luciferase in the presence of oxygen. This forms an intermediate compound oxidation reaction. Luciferase is oxidized, reacts with oxygen, releasing energy in the form of light. The reaction can be written as light emission, number of molecules, and the function of the growing fossilization within the growing molecules. When the light emission released during the reaction excites the molecules, which then return to their ground state by emitting photons, producing visible light, recycling. After the reaction, luciferase section remains unchanged and can catalyze another reaction, while the oxidized form of luciferin, oxyluciferin, is often recycled or excreted by the organism. This reaction is highly efficient, meaning most of the energy goes into producing light rather than heat. The ability to produce light gives bioluminescent organisms several evolutionary advantages. Attracting prey, predators like the anglerfish use their glowing lures to attract smaller fish, avoiding predators. Some organisms release bursts of light to startle predators or create a distraction. Finding mates. Fireflies use specific flashing patterns to communicate and attract mates. Camouflage. Some species use counter-illumination, producing light to blend with the faint light from above, making them invisible to predators below the surface of the ocean. Without awareness of their place in the cycles of life, without a flexibility to respond to their correctional situations, without a self-awareness of their luciferin and luciferase, none of these behaviors would be possible. There isn't just one universal form of luciferin and luciferase. Different bioluminescent organisms have evolved their own versions of these molecules. Here are some examples. Fireflies. Firefly luciferin emits a yellow-green glow when oxidized. Fireflies use this light to communicate and attract mates. Marine organisms. Many deep-sea creatures, like anglerfish and jellyfish, use sea lenterazine, a type of luciferin commonly found in marine environments. Their luciferase produces blue light, which travels farthest in water. Bacteria. Bioluminescent bacteria like Vibrio fischeri use a different type of luciferin and luciferase system. These bacteria often live symbiotically with marine animals, helping them glow fungi. Some glowing mushrooms use fungal luciferin, producing a greenish light. The purpose of this light is still debated, but it may attract insects to help spread spores. Each type of luciferin-luciferase system has adapted to suit the organism's ecological needs, making bioluminescence a versatile tool. The study of luciferin and luciferase has extended beyond the natural world into the field of biotechnology. These molecules are widely used in scientific research, medical diagnostics. Luciferase is used in diagnostic tests to detect diseases like cancer and infection. When luciferase interacts with luciferin, it emits light, helping researchers monitor biological processes. Drug development bioluminescence assays are used to screen potential drugs and study their effects on cells. Environmental monitoring. Bioluminescent bacteria are used to detect pollutants in water. If toxic substances are present, the bacteria stop glowing, signaling contamination gene on the cells. The blue tentacle passage, or Lee cells passage, engineering genetic engineering. Scientists have incorporated luciferase genes into plants and animals to study gene expression. For example, glowing plants have been created to demonstrate how genes are activated.